I'm David Hahn, and I will be starting my study aid. Alright. For my study aid, I decided to choose to do the role of the pancreas and liver in regulating digestion. And like I said, this is by David Hahn. So we'll just start off with the overview of digestion and small intestine, pancreas, and the liver, just because they're so interrelated with each other and how they function and just how they relate to each other. And so I'll just start off with the GI tract. It goes from the mouth all the way to the large intestine, but more specifically, the gut goes from the stomach all the way to the anus. And each food is passed to each compartment of the GI tract, and that the food that's passed down to each compartment is in the form of what's called chyme, and that's just pretty much just processed food that's mixed in with digestive enzymes. And chyme is pretty much passed to the small intestines from the stomach, and the stomach is where most of the mechanical digestion occurs. And so, because most of the mechanical digestion occurs in the stomach, most of the chemical the chemical digestion and absorption occurs in the small intestine. And because this is where absorption occurs, chyme moves through the intestines very slowly, just so the intestines are able to absorb as much nutrients as they can. And more than 3 liters of secretions are entered into the small intestines every day, and that includes digestive enzymes and bicarbonate, and those come from the exocrine pancreas. And secretions also include bile, which comes from the liver. And I have a picture right here that kind of shows where all 3 of the organs are kind of located in relation to one another. You can see the liver in the red, the intestines in the pink, and the pancreas right below the stomach, and they all have ducts that connect with each other, and those ducts secrete those digestive juices into the small intestine, or the duodenum, which is the first portion of the small intestine. Now we're going to talk about the role of the pancreas. The pancreas has 2 parts. It has an endocrine part and an exocrine part. And the endocrine part includes hormones such as insulin and glucagon. And these hormones are produced by what's called islet cells. And the exocrine pancreas, they secrete digestive enzymes like bicarbonate, and they are secreted by what's called acinar cells and duct cells. Acinar cells secreting digestive enzymes, and duct cells secreting bicarbonate, or NaHCO3. And these juices, they all enter into the pancreatic duct, and that's what leads to the small intestines. And so most pancreatic enzymes are what we call zymogens. Zymogens are inactive forms of enzymes that have to be activated once they enter the small intestines. And this activation process, it was described in Silverthorne as what's described as a cascade process that begins when the brush border secretes what's called enteropeptidase. And this is an enzyme that converts inactive trypsinogen into active trypsin, and this active trypsin then works to activate all the other zymogens and all the other enzymes that works to chemically digest the chyme. And there are certain signals that stimulate the release of these pancreatic enzymes, and they include, this distension of small intestines, or the movement of small intestines, the presence of food in the small intestine, other neural signals, and also the GI hormone CCK. Now we're going to talk about bicarbonate secretion, and we've seen this before in the past when we talk about the transfer of CO2, and bicarbonate is used to neutralize anything acidic, and in this case, the acidic chyme. And the cells that produce bicarbonate have high concentration of carbonic anhydrase, just like how we see it in red blood cells, as we learned before. And bicarbonate is pretty much produced from CO2 and water with the help of carbonic anhydrase, so it's kind of reasonable to think that cells should have a high concentration of carbonic anhydrase. And as that happens, the chloride enters the basolateral surface and leads through the apical surface through what's called the CFTR channel. and that chloride is then re-exchanged from the outside for bicarbonate, and that is how bicarbonate is secreted. And if these CFTR channels are very dysfunctional, it can lead to what's called cystic fibrosis. This is obviously with this dysfunctional CFTR channel, chlorine can't be secreted, and even though it's not can be secreted, mucus is still being secreted, so it kind of thickens and clogs up those pancreatic ducts, and that could lead to a lot of problems. And we could also see this in the lungs too, and that can also lead to some diseases or any lung infections as well. Now we're going to talk about the role of the liver and the gallbladder. Liver cells, or what being called the hepatocytes, secrete bile, and bile is pretty much a non-enzymatic solution that pretty much just helps digest fats. And bile includes bile salts, bile pigments, and cholesterol. And bile, travels from the liver to the gallbladder through what's called the common hepatic ducts. And once it's stimulated to release that bile, it travels from the gallbladder to the duodenum, which I said earlier is the beginning portion of the small intestines, through what's called the common bile duct. And the bile also does get reabsorbed. It gets sent back into the circulation. They're recycled back to the liver if it ends up, anywhere past small intestines like the large intestines. And, they're reabsorbed through the hepatic portal vein, which I believe we will talk about on this slide as we talk about the anatomy of the liver and the gallbladder. And we've talked about this before. The common hepatic duct takes bile from the liver to the gallbladder. The common bile duct takes bile from the gallbladder to the small intestine. The hepatic artery brings oxygenated blood from the peripheral tissue to the liver. And like I said before, the hepatic portal vein carries nutrient-rich blood from the GI tract to the liver. And so there you could just find, the reabsorbed bile and lots of other nutrients. The hepatic vein sends blood away from the liver back to the heart. And then we have what's called the sphincter of Oddi, which controls the release of bile and the release of secretions into the duodenum, just like any other sphincter would. Now we're going to talk about the digestion of fat by bile. And this process is pretty complicated just because, fat's not water soluble, and the fat droplets that we find in chyme are pretty big. So that kind of leads to less surface area for digestion to occur. And so in order to make it a little easier, we have what's called bile salts, which is found in the bile. And it's amphipathic. So it has both a hydrophobic and a hydrophilic region. The hydrophobic region interacts with the bile salt, while the polar side interacts with water. So that kind of creates a much smaller, much more water-soluble fat droplet. And now we can use the enzyme lipase, which is the main enzyme that digests fat. But because there is a bile salt coating, it cannot, or lipase can't penetrate the bile salt to digest. So we have to secrete what's called colipase, which is co-secreted by the pancreas. And with that, it is able to, easier digest the fat, make it a little smaller. And after the digestion of these fat droplets continue, they turn into what's called micelles. Now we're going to talk about how the fat is absorbed. And so as these fatty acids and monoglycerides move out of the micelles, they diffuse into the enterocyte through the process of simple diffusion. And once inside there, they go to the smooth ER in the enterocyte, where they combine to form what's called triglycerides. And from there, the triglycerides together with the cholesterol and proteins, they form what's called chylomicrons. And these are just much larger fat droplets. And then these are secreted through vesicles. These are packaged into packages by the Golgi apparatus, and they're sent out through the process of exocytosis and into what's called lacteals, which are lymph vessels of villi. And because chylomicrons are very large in size, it doesn't allow them to cross into cavities, but instead through the lacteals. And so they're sent right back into the venous blood right before it flows back into the heart. And I believe this concludes my study aid. Thank you.